Hello, welcome to Biology for You. In this video, we are going to discuss Griffith's experiment. In this series of videos, we have been discussing different concepts in molecular biology. In this video, we are going to have the basic idea of the Griffith experiment and what is the importance of it. So, basically, this was conducted in the year of 1920 time. During that time, many scientists wants to explore the physical basis of inheritance. That means how the inheritance patterns are happening from parental generation to the offspring. So, in order to determine the basic physical basis of the inheritance, many experiments were carried. So, it is one of such experiment which successfully described the process of transformation. So, the scientist named Frederick Griffith in the year of 1928. Frederick Griffith carried this transformation experiment in the bacteria called Streptococcus pneumoniae. This Streptococcus pneumoniae is a causative agent of the pneumococcal infection in the mice. So, the experimental organism he took is the mice. So, basically, Streptococcus pneumoniae is present in the two forms. One is R form and the other is S form. So, only the difference between R form and S form, that is two strains. R strain is having no polysaccharide coat around it, and this S form is having a polysaccharide coat . So, due to the presence of the polysaccharide coat, otherwise called as capsule, around this bacteria, it will be in smooth texture. And due to the absence of this polysaccharide coat or capsule around it, the bacteria will be present in rough structure, or it forms such colonies on the petri dishes. So, due to this rough shape, this is having irregular edges and it forms the irregular shapes in the petri dish, colonies of the petri dishes. And this smooth, which is having the polysaccharide coat around it, it will produce regular shapes and the colonies will be appeared in the smooth texture. So, that is the difference between R and S strains of the streptococcal pneumonia. So, basically, in his experiments, Griffith, he took R and S strains and he observed that R strain, due to lack of smooth or the polysaccharide coat around it, it is unable to cause infection in the mice, and hence it is called as non-virulent. It's a non-virulent strain basically, and due to the presence of S layer or the capsule around it, smooth strain is able to cause infection, hence it is called as virulent strain. So, due to the presence of capsule around this smooth bacterium, smooth strain, it is able to escape from the engulfment of the WBC, engulfment by the WBC of the mice cells. So, it is unable to, it will escape from this WBC or the white blood cells, and hence it can cause the infection. So, the mice that is infected with this S strain will eventually die. And the mice that is infected with the R strain will be survived or stay alive. So in his experiments, Griffith did one more thing. Along with these two kinds of strains, he used a combination of R strain, which is alive, along with the smooth strain, which is heat killed. So he used the R strain in combination with the heat killed S strain, and he expected that the mice may not die because this R strain is not virulent strain, and this smooth strain, which is present in the inoculum, is heat killed. It unable to cause the infection. But for his surprise, he observed that the mice, when it is infected with such combination, it is eventually developed the symptoms of pneumococcal disease, and it is dead. It was dead. And he observed that when he is using only R strain, it cannot cause infection, and when he use only smooth strain, that is S strain, which is heat killed, that is also unable to cause the infection. But if he use R strain along with this heat killed smooth strain, it able to cause infection. That means that this heat killed S strain is able to cause a change in the R strain. That means somehow the R strain is modified by the S strain, so that this R strain is eventually turned into a virulent strain, which caused inflammation in the infection in the mice, and the mice eventually died. So he concluded this as a transforming principle. That means there is something called transforming principle, which is exchanged from the S strain to R strain in this case, and this R strain is eventually turned into a virulent strain, and it caused pneumococcal infection in mice, and that leads to the death of the mice. So in the Griffith experiments, this transformation is explained. So there is an exchange of genetic material, or the transforming principle. He couldn't name the nature of the transforming principle, but he concluded the experiment with this, saying that there is something called transforming principle that is exchanged between the smooth strain and R strain, and from the smooth strain to the R strain, and then R strain eventually turned into a virulent strain. So that is what Griffith experiment is.